So thank you very much for this kind introduction. Thank you also very much for inviting me. And it's a great honor to talk here at the Turkish Seed Congress and about the future of Genebanks. I'm Manuela NAGEL from the Leibniz Institute for Plant Genetics and Crop Plant Research. We are located here in the middle of Germany. And I'm responsible or the leader of the research group Cryo and Stress Biology, and we work on conservation methods and deterioration mechanisms of seed, pollen, and clonally propagated plant. Here in this area, we have this very nice campus, and in the middle of this campus, there are the seed storage facilities. We have the documentation group, we have the cryo facilities, and also a huge herbarium. And I would like to go more in detail about this Genebanks and would like to structure my talk as follows. At first, I would like to talk about why and how do we conserve seeds. Second, what are the specific challenges and priorities for Genebanks? And third, I missed one topic, how to deal with these challenges. So come to the first topic, why and how do we conserve seeds? So, and with this, I like to go back in history several thousand years ago, and we come to the Fertile Crescent. I would like to exemplify this on barley and realize that you are here right in the middle of the primary gene pool where barley has been domesticated. This is already about 9,000 years ago where this happened. So, and barley was at this time then distributed over time. There centers of secondary domestication in North Africa, in Ethiopia, and also in Asia, which then moved back and forward so that here over time, also to America, a huge diversity established and barley adapted to all these different areas and regions and the specific needs. And you nowadays have actually a huge diversity of barley present. So you have the diversity in color, in shape, in awns, in the spike size, for example. Well, in general, you know even better than me the 2-row and 6-row types, all this diversity you have available and was used over hundreds of years until industrialization started. So in the middle of 19th century, the first more developed cultivars appeared and started to replace the old land races because they just had more yield or had a better stress resistant. However, so they started to replace this huge diversity of land races available, and several researchers realized this problem, and among those researchers was the Russian geneticist Nikolai Ivanovich Vavilov. So he started then to go for collection missions actually all around the world, and started to collect material, and he put it in the first gene bank in St. Petersburg. So that was at the beginning of the 20th century. Later on, other gene banks followed, so the IPK was established in 1943. Also, gene banks in the USDA followed at this time. And then in the 70s, a huge amount of gene banks were established because it was realized all around the world that our genetic resources start to disappear. And we have more than 1,700 gene banks around the world. However, that are data from 2010, so it's probably that there are more, or some are maybe also joined. There are international centers available, which you can see here. For example, the CIMMYT in Mexico, the CIP, the International Potato Center in Peru. You have the ICARA here, still located in Syria. Nowadays it's in Lebanon. You have ICRISAT in India. And you have, for example, also the International Rice Research Center in Indonesia. And then among that, you have quite huge facilities also available. The USDA has quite big gene bank. Russia is still a big gene bank. India has a own national gene bank, China, Japan, and you have the security backup here right in the north, the Svalbard Global Seed Vault. I will give you a bit more information about that a bit later on. Yeah, and among that, of course, also the IPK. So we are among one of the biggest gene banks in the world. To give that a bit, give you a bit numbers. So also from 2010. So we have 500,000 accession in the USA, around this in China. They still do a lot of collecting missions, so it's probably that they have increased their numbers already. There's about 360,000 in India, 300,000 in Russia, and as already mentioned, we are among the 10 biggest one with 150,000. There are these more specialized gene banks like the one here at IRI in Indonesia, which have just 11 genesis and 23 genotypes, so they focus really on rice. The same as, for example, here the CIMMYT in Mexico, they focus on maize and triticale, so wheat especially. However, here in Gaterslehm, for example, we have a collection of more than 3,000 different species, which has its own challenges, I have to say. So here in Turkey, I became also aware you have a quite big gene bank actually. So you have gene bank since 1964. It started with the Aegean Agricultural Research Institute, where it's called nowadays like that, in Izmir. And you have this there are more than 60,000 accessions, and you have this Turkish gene bank in Ankara with more than 60,000 accessions, and drying facility, storage facility, or equipment which is necessary for a gene bank. At IPK, we had also, as mentioned, the 150,000 accessions, and here you can see actually the gene bank, the seed bank manager. This is Andreas Börner. Some of you may know him, and he is also responsible for this big garlic collection of more than 23,000 accessions and the seed storage, as mentioned. Overall, we have 140,000 samples stored as seeds. So that means we do not have all the material stored as seeds. So some part of the material is still stored at the field gene bank because it doesn't produce seeds, or it cannot be maintained via seeds. This is the case for allium, for example, for the different diversity of garlic and shallot. Also for potatoes. However, potatoes are, for example, majorly maintained in slow growth storage under in vitro conditions. Those we have in our external collection in Groß-Lüswitz. And we have here in Gaterslehm a smaller collection of in vitro storage of mint, of Dioscorea, for example, and also some allium plants. We majorly focus here on cryopreservation to give these plants the chance to safely duplicate it in cryo. So, and currently there are 2,300 accessions, majorly potatoes, in cryo. So, in addition, just to give you an idea what is additionally done here at IPK, of course, the seed material needs to be maintained. So we have every year a field multiplication of about 5,000 accessions. We kept all the processes, the information about the protocols within the quality management system. We are certified via this ISO 9001-2015. And to compare our material, so to be sure that really this material, what we multiplied, is the same as we collected years before, we keep this huge collection of voucher specimens. So we have more than 450,000 voucher specimens in our storage in the herbarium. And in addition, we have also a collection of seeds, which is not kept viable, which is just for comparison, and a collection of spikes available that are 167,000 so-called reference samples we have here in our storage facilities. Yeah, and addition, so we follow the so-called ABS system. So the international standards for Genebanks, especially for seed storage, try to recommend the ABS system. So that means we maintain an active sample, a base sample, and a security sample. The active sample is used for distribution. So that's the one which you saw in glasses. In our case, it's kept at -18 degrees, also low moisture content. The base sample is vacuumized in aluminium foil bags, also kept at -18 degrees. And the security sample actually goes to the Svalbard Global Seed Vault. And currently we have more than 60,000 accessioned at the Svalbard Global Seed Vault. We just sent new, fresh multiplied material to the seed vault, and therefore it's increasing every year. And it's maintained there in their 3 storage chambers. At the moment they started to open the second chambers because the first one is filled, and will hopefully stay there for a long time until the material deteriorates and we need to replace this material. So now we come to the specific challenges and priorities of a gene bank. So according to Young B, and I completely agree with his priorities, the first priority of a gene bank, of course, should be the germplasm maintenance and reproduction. If you really need to focus because of a lack of money, this is the first what has to be done. The germplasm which is in the gene bank needs to be properly maintained and reproduced. And this is in addition or in combination also with data management. I'm aware that a lot of gene banks around the world have problems already to maintain the material, then data management already starts to be a problem. However, to document the passport data and the processes around this material is of highest importance for a gene bank. The second priority is then, of course, to make this material available for users. And so distribution is very relevant, and of course also acquisition, because most gene banks have the mandate to maintain also all national varieties which are not on the market anymore. So that's one main source for gene bank, the germplasm acquisition. However, also still collecting missions are important because we still have material around the world which are available in their centers of origin, in their centers of domestication, which needs to be collected. But here it's challenging to get the official permits to do so because we have the Convention on Biological Diversity, the Nagoya Protocol, and different regulatories from the countries which make the acquisition very hard, but it's still not impossible and very relevant to do so. So then there is the characterization and evaluation. And of course, this is a point which is very important for breeders, because if they like to choose material, they need to know which type of material they want that is available. So the data about that is very important, and of course also supportive research and the germplasm enhancement. And as I cannot focus on all the things around the gene bank and around the challenges, I would like to draw your attention to some aspects around the germplasm maintenance and reproduction, and the germplasm characterization, evaluation, and some supportive research according to that. So I come now to the germplasm maintenance and reproduction. The challenges which we have specifically here. And you have to keep in mind a gene bank collection is extremely diverse collection. And this is connected to a very specific management where you need trained staff. And this is the heart actually of a gene bank. People who are really aware which valuable material they have and which really want to multiply and maintain this material. And so in a gene bank like we are very happy to have the different curators who care for the different collections. And here you can see the lettuce collection, for example, which has to be grown, and then the challenges is to produce seeds. They are sometimes annual or biannual. You need also different equipment and different greenhouses for this kind of multiplication. So this is very challenging. If you have another crop like here, it's wheat. Of course, it has completely different growth dynamic, which you need to follow. So different curators are responsible for that. You can see here the harvest in the field. Here there is the harvest of some Vicia species, some legumes, which is again very different. Also the harvest procedure. You need to make sure that your material is clean. So a lot of gardeners has to clean the material over the year, that there is not a mixture also of the material, that you make sure that they are as less stress as possible during the vegetation period, so that you have finally a very high seed quality and a very high germination. Also, you need to know the system of multiplication, if it's a cross-pollinator or a self-pollinator. And of course, cross-pollinator have their own challenges, because you need to apply bee or flies or wrap them here in some foils to make sure that not other bee pollinate, for example, these sunflowers. And also, you have special systems for cereals. So if you have a diverse collection, it's very challenging to maintain and to multiply all material. And what you really need is trained staff. And this diverse collection also brings you to some other challenges. And this is related to seed longevity. So as soon as you have multiplied, you have harvested material, you have cleaned it, you made sure that you have really high quality seeds which are dried down, then you have the storage. And as I mentioned, we have the so-called ABS system, so active sample, base sample, security sample. And then we have, if there are still some seeds left, we have ambient seed storage, where we put the remnant seeds into to give them away to people who are interested for that in the first year after harvest. And with this material, we could do also some research, and that we did here. Actually, I did long time ago. So we investigated how long seeds can be stored under ambient conditions. And that was very interesting to see, because you have here, for example, brassicaceae seed, and you can see in the first years of reproduction, of course, they should have high quality, but then within few years, here in 15 years, the material dropped down, and you have here different accessions, which have also diversity itself. You can try to compare these different species by the so-called half viability period, and therefore I put these red lines here into. And a half viability period, or P50, is the time until germination drops to 50%. So you have here the 50% and here the time. So for brassicaceae, it's about 7 years. So for wheat, it's the same. It's very similar. You have also a time of about 7 years. But if you look into barley, I mean, we have focused on barley in between, it's a bit higher. It's about 8 to 9 years. And for pea, it looks like P is less diverse in their germination, and it can last much more longer in ambient storage. So you have here 15 years. However, of course, this is not optimum gene bank storage. So I would like to draw your attention also on this optimum condition, because when we put that at lower temperature, but very important is the lower moisture content, to continuously have a low moisture content, and the international rules say something between 15 and 35% relative humidity. Then you have here our barley collection, for example, which has been tested for with 12,500 tests, and you can see that still at the beginning there's quite a broad variation in the initial germination. This is considered to be a dormancy effect, so barley produces also dormant seeds. So it's a very challenging to find out the very initial quality, so you have to apply some dormancy breaking methods. And then over the years you have again also the diverse effect of germination of this collection. And the international rules say that you need to reproduce the material as soon as the initial germination is at 85% of the initial germination is reached. And of course, this can be quite quickly, so we have to deal with these challenges, and we have to also test it quite frequently. And it would be very nice to have also more faster viability tests available for in the future, especially for bigger gene banks like us. So, and just to complete this value we have here in barley, of course, under this optimum condition, a half viability of 155 years, whereas in brassicaceae it's 85 years, for wheat it's 150 years, and for pea 227 years. Of course, that are just estimated values, and it would be very great to know if the longevity can really kept for this long time under those conditions. So another challenge, so we had already the different challenges of diversity, of the different storage approaches, of the different species in the gene bank storage, and then we have another challenge that the years differ. So also we can feel climate change here in our area. But I tried to compare different results from different years, and I found, for example, for barley, it was that in 96, when the material was multiplied in 1976, we had a half viability period of 115 years. When the material was multiplied in 1975, and you may see it, it's not very visible, but however, on the P50 you can see it, the average P50 was then just 70 years. And when we multiplied the material, and here you really can see it, in 1974, then the P50 was just 50 years. So it really depends on the environment. So in every year there are some species which produce very high seed quality, and there are some species which doesn't like this condition at all and produce a very low seed quality. And we have to deal with these challenges and try to produce the highest seed quality we can. So however, in between all this multiplication and storage methods, of course, for breeders, it would be more important to have this characterization and evaluation data available. And this is a big problem because characterization and evaluation is time consuming. And I don't have this data from our gene bank, but it's available from the IRI, from the International Rice Research Gene Bank, and they have actually, I hope you really can see that, half of this collection are land races actually, and the other half of this collection are improved varieties, and they data, how they characterize their material. And the blue one is 89% of this material has been already characterized. So there are specific descriptors like flowering times, like plant height available. And according to this descriptor, the whole collection has been characterized, what is extremely valuable. However, when it comes to traits where breeders might be interested in, for example, like grain quality evaluation, then you can see most part is red. So 66% of the collection has not been characterized. And only, for example, for drought and cold stress or something like that, only 3% has been characterized. And for other abiotic stresses like diseases and insects, only 12%. And this is probably very similar here at IPK. So to do the characterization and evaluation is very time consuming, and it needs specific projects. So this is a challenge definitely for the future. However, how can we deal now with these challenges? And I would like to come back to the germplasm maintenance and reproduction again. And I must tell you concerning the problem with the seed viability testing, there are already some solutions out. So, for example, the International Rice Research Center, they started to initiate or to establish a lemna tech system where they do high throughput phenotyping or high throughput germination of their whole collection, and very efficient. However, the problem is for us, for example, I'm not so sure if that would be that efficient, because we have a collection of more than 3,000 species, whereas the IRI has a collection of 24 species. So they have more homogeneous genotypes. And for us, the challenge is to really adapt this to the specific needs of the species. However, I think that may come in future. There are also other possibilities out. And in the last years, we also tried to look into this non-invasive viability monitoring, if this makes sense, if this is useful for us. To give you an overview, there are, for example, different systems available. There's machine vision system. Here you usually have the spectrum of visible light. You have a camera system, and then you get data like you can do the seed grading, you get data like color, shape, area, and usually this is analyzed via machine learning system and is well established already in seed industry. Other systems are spectroscopical methods. So you have the near infrared or the mid-infrared system, or the Fourier transmission infrared system, or the Raman spectroscopy. And for near infrared and mid-infrared, it's usually you have a source, here a source available. This emits the certain in a certain wavelength, and the sample pushes back a certain wavelength which is collected. The information are collected by a detector and analyze them, and you can get quite a lot of quality measurements around that. For example, NERS is quite well established for protein content, for water content, for example, and also other composition major composition components they try to cover. Similar also Raman, you have here also laser and detector, which gives a specific wavelength, and you can try to get data about the specific seed quality. Other methods are, for example, hyperspectral reflectance. Hyperspectral means that you have very broad range of wavelengths available, which you analyze via DeepL, so via machine learning approaches. And at the end, if you look into the papers, you get correlation coefficient for single genotypes or several genotypes of 98%, so predictiveness for germination. So 88 to 100%. Can this approaches predict germination, or 84 to 100%? However, as I mentioned, this is usually single genotypes, and it's very hard to predict the germination for a whole collection. But I'm quite sure that there will be improvements in future. What we tried here at IPK as well is, for example, also to analyze the metabolite diversity and to come to a conclusion based on these results. So I analyzed here a mapping population of 184 genotypes and did a metabolite analysis, and the highest correlation which we found is for butanoic acid. There you have 60%, so the higher the germination, the lower the butanoic acid content. It's very similar for gluconic acid, and for glycerol, that's over several species, a strong indicator for a lower germination. So the higher the glycerol content, the lower the germination, which indicates the lipid deterioration. Yeah, and adenine we found as well. However, you still can see the germination correlations are not very impressive. So for the future, I believe that a combination of this method is required to come really to a predictive germination results and to a high throughput germination test in Genebank. So another priority, another challenge which I mentioned before, is the characterization and evaluation. And I think we made a huge step forward in the last year. Here at IPK, the sequencing and genotyping approaches were massive step forward, and this is headed here by Martin Mascher and Nils Stein at IPK, and they started to genotype the whole barley collection, all the 22,000 accessions which we had in our storage. And for example, you can see here it was genotyped by GBS, so genotyping by sequencing, and you can see the via this PCA where the material comes from. So for example, from China, from India. When you look here into this map, most of the material, of course, came from Asia and Northern Europe, but you find also material from Ethiopia, and here also from the fertile questions here in green, also from Turkey and this area. So that really shows you the diversity of this material. And this is only exemplified for barley at the moment. Wheat is genotyped, the rye collections, the bean collections. So we continue with the different collections to characterize and genotype them by this approach. And the value of this approach is at the end that it supports duplicate finding. So we can focus on specific material and develop only specific material. It helps to identify unique genotypes and genes which are relevant then for breeders later on, and that we are able to do a kind of compilation of subsets for further evaluation. This is of high importance then later on for really the usability of this material. And another step forward is that we nowadays can also make sure that this data are very available. We try to follow the FAIR principle, so findable, accessible, interoperable, reusable. And these, for example, the barley data are available via the Bridge portal, and you can choose around from this 22,000 accessions, can get this genotyping data and also few phenotypic data which have been made available. And another huge step forward is that then we can go to the really precision plant phenotyping. And here at IPK, but also in other research institutes nowadays, LamnaTech technology, LamnaTech phenotyping is available. And nowadays we started also with the so-called plant cultivation hall, which gives you a new idea of precision phenotyping. And I'd like to show you a short video which gives you an idea what the plant cultivation hall might be able to do.
A highly innovative plant cultivation facility is currently under construction. The Plant Cultivation Hall. It is specifically designed for high throughput phenotyping, with plants growing under conditions that are typical to the field, but which can be imposed in a highly reproducible manner. Unlike in field trials, environmental conditions such as light input, air temperature, CO2 concentration, and air humidity, as well as water and nutrient supplies, can be precisely set and regulated. The Plant Cultivation Hall will comprise 2 different phenotyping systems in an area of almost 500 square meters. The Rhizotron system is being developed and implemented in collaboration with the Forschungszentrum Jülich. It will support the simultaneous cultivation of up to 360 plants and will enable very detailed studies of the root architecture. The other area of the hall houses the container system for the phenotyping of stands of plants of vegetation. Various camera and sensor systems are mounted on a robotic crane that can be precisely positioned. These plant imaging systems use different wavelengths and scan them in 3 dimensions. The soil in the containers resembles the natural soil structure in the field. Its temperature can be precisely controlled at different depths. The temperature and soil moisture are monitored by numerous sensors. Built-in imaging tubes.
So this facility is very great to analyze our subsets, to analyze our core set, and do really precision phenotyping. And then finally, I would come to our research on seed longevity for the future. And here I'm very happy that I'm a part of a team of 7 Genebanks actually. So together with the Norgene who manages this project, the Embrapa in Brazil, ICRISAT in India, INIA from Portugal. So we are a team who really are interested about the longevity of material at the Svalbard Global Seed Vault. And what we try to do is to bring all the aspects of seed longevity together to analyze diversity, and therefore we have 13 crops included in this experiment, among also pea and chickpea, maize and other cereals like rice and maize and barley. And not only the species diversity, also the environmental diversity. So we will use 3 environments of reproduction, and the genotype diversity. So we will use 5 genotypes per species to come to a conclusion. And of course, this material is distributed via SMTA, including all passport data, including all information of multiplication, then stored at IPK. And finally, we will do frequent germination tests over 100 years of storage. So this is really an experiment which is done for the future, Genebanks, to get more information about this material available, what we have in Genebanks. And with this, I like to also conclude my talk now. So I hope I covered now some aspects about Genebanks. So why are we doing that? What are we doing exactly in Genebanks? I'm aware that a lot of Genebanks are heavily underfunded and have to deal with the first priority, the maintenance and the reproduction of the material. But I hope that most Genebanks can have in future trained staff and the technology, the equipment available to maintain a huge and genotyped unique collection, which is then securely long-term stored, which is evaluated and characterized, and which is used for germplasm improvement and to finally ensure the food quality for future generations. And with this, I like to thank, of course, my co-workers here at APK, especially Andreas Börner. Another one is my colleague from the University of Innsbruck, which is especially Ilse Krane. Some data came from the so-called EU EcoSeed project. And thank you very much for your attention, and I'm open for further questions. I have added also some references, and I would like to draw your attention finally to the Seed Symposium, to the ISTA Seed Symposium. I'm responsible for the organization of this ISTA Seed Symposium, and this year, next year, it's supposed to be in Christchurch, but unfortunately, due to restriction, we had to postpone it. This will be in 2025. But we try to organize a seed symposium in the next year. And so far, try to keep updated if you are interested in this. This probably, so we are still in the planning phase. We want to have the meeting in November 2022. At the moment, we are looking for the location in Greece. It will be probably a symposium of 2.5 days, and we want to integrate also Young at ISTA, who will organize a half-day symposium and a conference tour. So please stay tuned, try to keep updated. There will be the ISTA Seed Symposium next year, but not in New Zealand, in another European country, and that will be most likely Greece. And so for this, I'd like to thank you very much.